Good evening. I want to thank everyone for braving the cold and coming in to what I think is going to be a very exciting evening. The university lecture, which was inaugurated in 1950, is one of the most enduring traditions of Boston University. It's also one of our most interesting. This annual event embodies our mission as a leading research university, and importantly, it shares the very best of the university's research and scholarship with the public. It is a celebration of the best of a research university and the best of Boston University. This year's lecturer, Professor Karen Warkentin, is a wonderful selection for this lecture. Her research started with some very small creatures living far away, tree frogs, in the rainforest in Costa Rica. Because Dr. Warkentin happens to be good at thinking big, her field of study expanded from tropical frogs to humans. It got there by way of scientific revelations about animal sexuality. Her findings and her teachings have evolved in parallel with social changes that have increased the visibility and acceptance of diversity in human sexuality. Since 2011, half of her teaching has been in our Women's Gender and Sexuality Studies program. The implications of her current work are relevant to everyone in this room. Her research results lead to questions about what we are, what we think we should be, and what we could be. Dr. Warkentin's research and teaching are extraordinary for their depth and their breadth, incorporating multiple disciplines and new ways of seeing and thinking. Perhaps that should be expected from someone who grew up in Kenya and Canada. Professor Warkentin earned her bachelor's degree at the University of Guelph, her master's degree at Dalhousie University, and her PhD at the University of Texas at Austin, the easiest of the three to pronounce. She also did postdoctoral work at the University of Kentucky and the Smithsonian Tropical Research Institute in Panama. She is a world traveler. Her research is changing our understanding of how life works. She has always painted on a broad canvas, and she has never been afraid to paint outside the lines. That's one reason that her thinking and her teaching encourage all of us to reimagine the potential and variability of living things, from tropical frogs to ourselves. Her lecture tonight is entitled Diversity and Plasticity of Life, a biologist's journey from embryo self-defense to sexual behavior. Karen, the podium is yours.
Thank you, President Brown, for that wonderful introduction. Thanks to the University Lecture Committee for inviting me to give this talk, and thanks to you all for coming. I want to share with you tonight some highlights and perspectives from my work on diversity and plasticity. So I'm going to tell you first 3 stories from my egg science research, or lessons the frogs have taught us, about embryo self-defense, reproductive plasticity, and family diversity. Then I'll look beyond frogs to consider sexual diversity in animals and people. So I didn't start out as a new PhD student planning to study embryo self-defense. Its discovery emerged from basic natural history, which raised questions, generated hypotheses, and led to experiments. But what I learned has shaped my entire research trajectory, from my PhD work through my current work with students and collaborators, and essentially created a whole new field of study on environmentally cued hatching, changing how we understand embryos, from eggs developing in preparation for their future life to organisms interacting with and responding to their environment. My work is part of the renaissance of research in the larger field of phenotypic plasticity. And plasticity is the capacity of genetically similar organisms to develop differently or express different traits in response to different environments. It is a ubiquitous property of life, and it affects all sorts of organisms and all sorts of traits. So the context-sensitive nature of development means that all individuals express only a subset of their potential and contain a rich array of hidden possibility that might be expressed in some other context. Plasticity means that genetically similar organisms can look and act different depending on their past environments, but also that genetically different individuals may respond similarly to some environmental contexts and differently to others. Thus, there's no one-to-one relationship between either genetics or environment with organismal traits. Rather, there are suites of sometimes complex interactions between many factors. Moreover, individuals that seem very similar may have reached that state by different mechanisms through different developmental trajectories. Through my career, work on plasticity has progressed from simply documenting responses and assessing their adaptive function to determining the detailed mechanisms underlying their regulation and examining their larger implications. This work has fundamentally changed our understanding of how life works, from the mechanisms that regulate gene expression within cells and how these can scale up to affect ecosystem processes, to the mechanisms that enable evolutionary change and what happened in the history of life. My work on plasticity started with frogs, or more specifically, with frog eggs. But if I had been a different person, I would never have discovered escape hatching. So my advisor at the University of Texas was studying these frogs. His work focused on sexual selection, so how males compete for females and how females choose males as mates. It is famous work, and he had funding to support it, so most of his students chose projects in that area. But those were not my questions. As a queer young biologist, I didn't want frog heterosexuality to be the focus of my PhD. I was really much more interested in matters of life and death, predation and escape. And I wanted to study early life stages whose sex was neither obvious nor really relevant to my research. So I needed to find my own question. So in the summer of 1991, I went to Corcovado National Park in Costa Rica looking for a frog with interesting eggs or tadpoles and a question for my doctoral research project. I was introduced to the beautiful red-eyed tree frog, which lays terrestrial eggs on leaves over ponds, and at hatching, their tadpoles fall into the water, where they'll develop until, at metamorphosis, they climb back out of the water and up into the trees. And I started monitoring their egg clutches, doing basic natural history at this pond. All these little pink flags mark egg clutches. And as you can see, there were a lot of them, sometimes even two on one little leaf. I soon learned that a lot of eggs were eaten by snakes. In fact, snakes ate half of the eggs that I was monitoring at this pond. And if we think about our own relationship with eggs, maybe it's not surprising. Eggs are nutritious, delicious, and if they don't have parents around to protect them, pretty much helpless. Or so we thought. We don't usually think of eggs as doing much, except developing, of course. And most biologists who study eggs work on development in controlled laboratory settings. But one thing all eggs must do eventually is hatch. So one day at the pond, I accidentally bumped into a well-developed red-eyed tree frog egg clutch and saw a few embryos hatch out and fall into the water below. And it really surprised me how fast it happened for what was supposed to be a slow, fundamentally developmental process. So thinking about these two things, I came up with a hypothesis of adaptive plasticity in hatching. I thought maybe embryos use hatching as a self-defense strategy, rather than just sitting there and letting the snakes eat them. But if they can hatch earlier, why would they wait around when they might be attacked? There would have to be some trade-off. So I hypothesized that predators in the water might impose natural selection against hatching early. If less developed tadpoles were more likely to be eaten, and more developed tadpoles that hatched later were better able to escape. When I presented these hypotheses, along with my egg predation data, 50% eaten, at a conference, I encountered some extreme skepticism from some senior colleagues. Paraphrased here. People thought it was impossible for frog embryos to hatch fast enough to escape from a snake. And other frogs were known to take many hours or even days to slowly weaken and eventually rupture their egg membranes by enzymatic digestion. People also thought it was impossible for a few days of embryonic development to actually affect tadpole survival in the water. And they advised me against wasting my time on a project doomed to fail. I wanted to test my hypothesis. So I went back to Costa Rica and I let snakes attack eggs in cages where I could watch them. And I saw embryos hatch and escape in my cages. And I also documented escape hatching at the pond. I found that the pond was full of these freshwater shrimp and that older hatchlings were indeed better at escaping from them and from other aquatic predators. When I submitted my work for publication, one reviewer commented, "Every herpetologist knows that if you jiggle well-developed frog eggs, they hatch. It doesn't mean anything." They seemed to think that the snakes were just somehow inadvertently breaking weakened egg capsules, releasing the passive embryos. It couldn't possibly be the embryos saving themselves. So I'm going to show you a short video, and you can judge for yourselves. So these embryos are 30% premature. The snake has already eaten 2 eggs, and these 2 have ruptured their membranes, beginning the hatching process. I'll show you the 3rd bite, and for this embryo, you're going to see the whole process in real time, from decision to escape. Okay, so here comes the snake, jiggling, biting the eggs. See the embryos wiggling? Okay, this one's going to pop out. There he goes. Okay, and watch this one starting to wiggle. It's decided to try to hatch. And you see the leak happening, that change in reflection. So it's made a hole. It's going to poke its little nose through there. Oh, a little hard. Got out. Okay. It escaped. I never get tired of watching this. So I think predator-induced early hatching is actually a great example of phenotypic plasticity. And I hope you're convinced that it can serve as a mechanism for self-defense, getting away from a snake. The editors were convinced, and my first paper on escape hatching was published in 1995. This work then motivated and inspired many other scientists around the world to do related studies. And so we now know that environmentally cued hatching is actually widespread. It's been found in all 15 families of amphibians where people have looked for it. And it's been broadly documented in other animals, from flatworms and mollusks to birds and mammals. Embryos respond to risks, and they respond to opportunities. Just like later life stages, embryos are subject to natural selection. They gather information about their environment, and they respond in ways that improve their survival. It seems kind of obvious now, but it didn't used to be. So knowing that embryos can actually flee from danger raised many new questions. How do they hatch fast enough to escape? How do they know when to hatch? How does development affect their escape hatching behavior? Such questions still motivate my research. So since 1998, my fieldwork has been based at the Smithsonian Tropical Research Institute in Panama, and I've taken many students and collaborators there over the years. Together, we've learned that embryos hatch to escape many dangers, not just snakes, but also egg-eating wasps and pathogenic fungus, and physical threats such as flooding or drying. We also documented multiple costs of hatching early and how these vary among aquatic environments with different predators, changing the future prospects of individuals that hatch at different ages. We studied how these embryos hatch, since it's clearly not just a passive developmental process. Frogs had been described as gradually releasing a hatching enzyme that over many hours or days broke down the whole membrane until eventually it released the embryo. When red-eyed tree frogs decide to hatch, they push their little nose against a single spot in the membrane, digest a small hole, and then leave, leaving the rest of the egg membrane unchanged. So my student Christina Cohen found that the glands that make and store the hatching enzyme, these little spots, are highly concentrated on the embryo's snout here. So it can be released at one spot on the membrane. Taking a closer look at the cells, she found that they store up a large supply of hatching enzyme, and then they dump it suddenly in bulk, allowing the embryo to actively regulate when it hatches. So Christina's discovery of this novel hatching mechanism in red-eyed tree frogs shows that frogs don't all hatch the same way, and those differences matter. Other frogs also have long plastic hatching periods and rapid responses to threats. But we don't know if they've converged on the same solution or if they've come up with different ways to speed their escape. Now, of course, a rapid hatching mechanism is only part of the story. Embryos also need information to decide when to use it, and egg jiggling and attacks seem to be key. So since 2001, I've collaborated with engineering professor Greg McDaniel to measure vibrations in egg clutches and to study how embryos use them to assess danger. So since predators can't eat eggs without moving them, there are always vibrations in attacks. But many benign forces, like torrential tropical rainstorms, also jiggle eggs, creating a risk of false alarms. Making matters worse, to escape from a snake, embryos have to decide pretty quickly, but the structural mechanics of clutches makes eggs jiggle in similar ways in response to any kind of force. Nonetheless, the embryos are very good at identifying threats and avoiding false alarms. How do they do it? So by shaking eggs in controlled ways, using a whole variety of devices that we have designed for this purpose over the years, we've learned that embryos actually combine information from many different vibration properties to assess risk. And this actually also tells us something important and more general about how animals can use messy information to make important behavioral decisions. This is a common problem in predator-prey interactions, and it's hard to study, but with red-eyed tree frogs, we can use vibration playback experiments to get at this. We're now exploring how developmental changes in what embryos are able to perceive as their sensory systems develop, and on what they optimally should do based on changing risk trade-offs, combine to shape their decisions at different stages. All animals, like people, behave differently at different ages, different developmental stages, for lots of reasons. And these embryos are really beautiful for studying this, since so much about their lives changes so dramatically over just a few days. And I'm sure that red-eyed tree frog embryos still have a lot to teach us. But we can also learn from other frogs. My first PhD student, Justin Touchon, studied hourglass tree frogs. These frogs are common in the same ponds where red-eyed tree frogs live, and they independently evolved to lay terrestrial eggs. Like me, Justin began by monitoring egg clutches at rainforest ponds. He found that eggs died a lot from drying and from predation, and he saw embryos hatch early to escape from both threats, as in red-eyed tree frogs. And then one day, Justin decided to collect eggs from a pond in an old gravel quarry right in town. It's more open and hot than the nice, shady rainforest ponds, and so we didn't usually work there. But it had great choruses of hourglass tree frogs, and it's super conveniently located. So typically finding hourglass tree frog eggs is easy, and Justin was an expert. But at that pond, he couldn't find any eggs. Okay, so this is super weird because we knew there were lots of the frogs there. So we went back at night to follow the frogs and solve the mystery. And we saw Justin's frogs laying their eggs in the water. Okay, this was shocking. Frogs laying eggs in the water may not seem shocking to you, but this was shocking. Because as tropical frogs go, right, this species have been studied a lot, and they had always been described as a terrestrial breeder. And they were at nearby forest ponds, but apparently not at Quarry Pond. What is going on? So we knew ancestrally frogs laid aquatic eggs, as they do, here in Massachusetts. But they've evolved to lay terrestrial eggs at least 50 times in evolutionary history. But these transitions happened a long time ago. And to our knowledge, all living frogs had either aquatic eggs or terrestrial eggs, not both in the same species. And indeed, this dichotomy seems to stem from a fundamental physiological trade-off. Most terrestrial eggs don't get enough oxygen underwater, and aquatic eggs dry out and die if they're on land. Our discovery of aquatic and terrestrial reproduction in the same species meant long-standing hypotheses about how terrestrial eggs evolved could actually be tested experimentally. Opened up all sorts of new research possibilities. So Justin first asked if the variation between frogs at different ponds was genetic or if it was a response to their different environments. He caught frogs at both sunny and shady ponds, and then he put some into kiddie pools in shady environments and others into kiddie pools in sunny environments to see where they would lay their eggs. And what he found was that all the frogs, regardless of where he caught them, made a plastic behavioral choice about where to lay their eggs. These frog parents decide where to put their eggs based on the risk that the eggs will dry out on land. And in another experiment, Justin learned that they also evaluate the risk of egg predation in the water. So both of these threats to egg survival vary among nearby ponds, imposing opposite selection pressures on the eggs and on the frog parents, which makes egg-laying plasticity an adaptive strategy. So these embryos can develop normally both in water and in air. While they are at greater risk of drying than more specialized terrestrial eggs, they're at much lower risk of suffocation underwater, giving their parents a lot of flexibility. So in Justin's continued work on reproductive plasticity, pursued as a Smithsonian postdoc and now as a professor at Vassar, he found another 5 species closely related to hourglass tree frogs that plastically lay eggs on water or in land, and they're marked in orange on this family tree. He found other related species whose eggs are always aquatic, marked in blue, and others where the eggs are always terrestrial, marked in green. He also found 2 populations of hourglass tree frogs where the eggs are always laid on land with no parental response to risk of drying, suggesting that they're genetically differentiated. And more recently, he's found individual variations in the egg-laying preferences of different frogs here, and he's breeding them in the lab to study the evolution of reproductive mode. So Justin's work suggests that plasticity is an evolutionary intermediate that can form a bridge between these 2 more specialized, less flexible states. Our discovery of reproductive plasticity in a lineage of frogs that repeatedly evolved terrestrial eggs has created new possibilities for experimental research to understand how this happens, changing our understanding of evolutionary history and the kinds of questions that we can ask. My most recent PhD graduate, Jessie D'Elia, works on glass frogs. These beautiful little frogs live along forested streams in Central and South America, and they independently evolved terrestrial eggs. Unlike hourglass tree frogs or red-eyed tree frogs, some glass frog parents actually care for their eggs. And when Jessie started working with me, we thought that about 20% of species had parental care, and it was always by fathers. So these fathers collect water and deliver it to their eggs, and they also guard them from predators, small predators. In his master's work in Mexico, Jessie had discovered that embryos that are abandoned by their fathers hatch early to escape from deteriorating conditions in the egg clutches and embryos whose dads stick around develop for much longer in the clutch with that continued care. But really, we knew very little about even basic natural history of most glass frog species. So Jesse, like me, like Justin, started with fieldwork, walking along streams at night, watching frogs, monitoring egg clutches, and he actually spent years in the field. He worked at 40 different field sites, 40 different species and 22 field sites across 5 countries, partly in collaboration with Laura Bravo Valencia, who was at the time a student at the Universidad de los Andes in Colombia. And what Jesse and Laura saw dramatically changed our understanding of glass frog families. They confirmed previous reports of male care, and unsurprisingly, they documented additional similar cases. But in one species, Ikakogi tyrona, they found mothers looking after their eggs, showing behavior very similar to males of other species. And you can tell this is a female because she's a glass frog, and you can actually see her eggs, her next clutch of eggs, through her side there. In addition, in many species that had been thought to lack care, they found females, mothers, sitting on their egg clutches for hours after laying them. Now, these mothers initially seemed really dedicated, but they never stayed beyond the first night. So Jesse and Laura tested if that little bit of care actually made a difference for egg survival. They found that it was essential. In just a few hours of brooding, mothers transferred a lot of water to their eggs, enabling embryos to survive for days without rain. Moreover, the hydrated egg jelly protected embryos from predatory insects, which had difficulty chewing through that swollen jelly, and no difficulty eating eggs that had not been brooded. So this new evidence that all glass frogs have and all glass frogs need parental care changed our understanding of sex role evolution. Previously, it seemed that male care had evolved twice from ancestors with no care. Now it appears that male care evolved 3 times from ancestors with female care. Surprisingly, each time that males took over caring, they did more for their eggs and they stayed with them for longer. Even Ikakogi mothers, who stay for many nights, abandon their embryos before they can hatch. Some fathers continue caring for many days or even weeks after their embryos are able to hatch, allowing those embryos to develop more before hatching, which improves their survival in streams full of fish. And Jesse found that how long fathers care for the eggs is directly related to how many egg clutches they have. Both across species and within species, fathers who mate more, stick around, and care for their eggs for longer, sometimes for months. Moreover, evolutionary increases in care duration are associated with increased plasticity in hatching timing, enabling embryos in species with potentially excellent fathers to stay even longer in the safety of a well-cared-for egg clutch, but without curtailing their ability to hatch early if they need to. So these findings on minimalist female care and elaborate, prolonged male care contradict common ideas about how sex role evolution works. Conventionally, males are thought to face a conflict between caring for offspring and seeking new mates, and that is thought to limit paternal investment. Clearly, glass frogs do not accept that premise. And work in other animals has revealed that sex role evolution is actually far more diverse than predicted by conventional theory. In frogs, male care and female care seem to have evolved about equally often, creating opportunities to study sex role evolution and parental care in a sex-balanced context. And so Jesse's work contributes to a growing body of research suggesting that the extreme of female-biased care that we see in mammals may be more of a mammalian peculiarity than an evolutionary generality. So my discovery of embryo self-defense and subsequent work on environmentally cued hatching changed our understanding of animal embryos more broadly. Justin's work is changing our understanding of how terrestrial reproduction evolves, an event that clearly happened in our distant past. And Jesse's work on elaborate paternal care and minimalist mothering contributes to an ongoing shift in our understanding of animal sex roles. Each of these discoveries required noticing things that were not supposed to happen, taking our observations seriously, and then doing the work to test the hypotheses that came out of those observations. They involve spending time in the field, of course, but I cannot have been the first herpetologist to bump into a red-eyed tree frog egg clutch and see embryos hatch, nor can Jessie have been the first person to see a female glass frog sitting on her eggs. But different people notice different things, think about them in different ways, and are more or less strongly motivated to pursue them. A lesson the frogs keep teaching us is that animals don't always do what they're supposed to do. And when what we think we know conflicts with what the animals we study do, the animals aren't wrong. We are. And this mismatch of received knowledge with direct observations of natural history and behavior creates new research opportunities, and it can lead to major shifts in understanding. So in my career as a biologist, I've also seen dramatic changes in how we understand sexual variation from both evolutionary and developmental perspectives. But these changes are not all widely appreciated in society. So since 2011, I've taught this work to women's gender and sexuality studies freshmen, some of whom are here today, and also to biology graduate students, ditto. I want to share with you some key points from this that I think are useful for understanding our own diversity and life more broadly. Okay, so we used to think of sex determination, becoming male or female, as a simple kind of switch and cascade. Now we know there are complex interacting gene networks at play and many ways to tip the balance toward male or female, and this can be rebalanced even in adulthood. We know that even at the most basic levels of genetics, gonads, and genitals, sexual development is neither completely dimorphic nor completely consistent across levels, which means that not all people fit clearly into male and female categories. This variation is a spectrum, and there's debate about who counts as intersex, but the best current estimate is about 1.7% of people, making intersex people about as common as natural redheads. Other kinds of sex-associated traits vary even more. So individuals, rather than being uniformly feminine or uniformly masculine, express unique combinations of feminine, masculine, and intermediate traits. For instance, here, each column shows the state of the 10 most sex-differentiated regions in brain scans of a set of women and men. And you can see that there are general patterns that could come out statistically, but there's no uniformity and there's no internal consistency. In keeping with our current understanding of sex-specific traits as different expressed subsets of shared underlying potential, cross-sexual transfer of traits happens frequently, simply by changing expression patterns. So, for instance, in some species, like these hummingbirds, there's a clear sexual dimorphism, but there's also more than one way to be female. So the magnitude of sex differences depends on which subset you compare. Similarly, orangutans show a striking sexual dimorphism, and these flanged males are twice as big as females, and they have these dramatic enlarged cheek pads. But there are other males, also reproductively active, that pretty much look like females and are the same size as females. And in bluegill sunfish, there are clear differences between large, showy, nest-building males and the females that they spawn with, who leave them to care for the eggs. But there's also males who look like females and spawn in threesomes with females and the large males. And there are actually many well-studied examples of such female mimic males, and our understanding of male mimicking females is also growing. Moreover, the striking sexual dimorphisms that we see that are very evident in some species are completely absent in other closely related species. And of course, cross-sexual transfer of traits also happens with behavior. For instance, either mothers or fathers or both can care for young. And this has switched many times in evolution. So remember Jesse's frogs. We can think about same-sex sexual behavior and pair bonding in this context as cross-sexual transfer of behavioral traits, involving either what the animal does, who it directs its behavior toward, or both. And our understanding of animal sexual behavior is an essential frame for how we think about human sexual behavior and what kinds of questions we ask. Unfortunately, this understanding has been hampered by denial, neglect, and trivialization of observations that didn't fit with what was supposed to happen, like we saw with the frogs. The animals, however, continue to do what they do. Some field biologists, finding that same-sex sexual behavior in their study animals was simply too common to ignore, have produced substantial research focused on understanding its functional relevance. And this work has accumulated in parallel with increasing visibility and acceptance of diversity in human sexuality, profoundly altering our understanding of sexual behavior in animals. I think it's also useful for understanding ourselves. So the classic view of animal sexual behavior was that its sole purpose is sexual reproduction. And with this framing, some human sexual traits were deemed special and others unnatural anomalies. A modern comparative perspective suggests neither is correct, freeing us to consider human diversity in evolutionary context. So first, although many animals mate only when reproduction is possible, in others, non-reproductive sexual behavior is common. For instance, in over 130 mammal species, females engage in far more sex than is needed for conception, including when they couldn't possibly conceive, and including with multiple males. So it's not just humans who can do this. In about 80% of these species, including the primates, the best explanation for this apparently excessive sexuality is that it originally evolved to confuse paternity and protect vulnerable young from potentially infanticidal males. When this happened, mating took on a new function beyond simply the union of egg and sperm. Also, at least for females, the physiology of attractiveness and the neurobiology motivating sexual behavior also must have changed. And this opened new possibilities for other evolutionary changes. Among primates, in our most distant cousins, the lorises and lemurs, sex is focused on reproduction. It's confined to a short estrus period when they can conceive, and it's strongly hormonally regulated. It's just not that interesting if conception is not possible. In contrast, Old World and New World monkeys and apes use sexual behavior for many social functions, not just reproduction, not just paternity confusion. Hormonal control of sexuality is much weaker. For instance, even females without ovaries can remain sexually active. And it seems that an ancestor of these highly social species, perhaps 45 million years ago, co-opted behaviors that were originally reproductive, just reproductive, to serve additional social functions, such as affiliation, dominance, reconciliation. Today, their descendants still use sex for social functions, and they also engage in it simply for pleasure, since once it evolved, the physiology of orgasm works even without direct fitness benefits. This kind of change where a trait that originally evolved for one function is later co-opted for another, is really common in the history of life. And once sexual behavior had evolved to do more than just enable the fertilization of eggs, it would have come under selection in new social contexts. So both its uses and its expression expanded substantially. Of course, non-reproductive functions of sexuality need not be restricted to male-female interactions. And in many primates, they are not. The most familiar example is probably our close relative, the bonobo. These apes use sexuality for all sorts of social functions, including conflict resolution, stress reduction, and relationship building. And it seems that bonobos, all bonobos, interact sexually with both males and with females. And same-sex sexual behavior has been documented now in a great diversity of species. It takes many different forms, ranging from indiscriminate mounting or temporary misidentification of partner sex to enduring, multifaceted sexual and social bonds. It's estimated to occur in 15 to 30% of mammals and birds, and it's particularly common in highly social species, where there's lots of opportunity for frequent and varied social interactions that are important for individual success. In most such species, individuals interact sexually with both males and females, not exclusively with either sex. So if we take another closer look at primates, same-sex mounting appears rare or absent in lorises and lemurs. It's been documented in 3 genera of New World monkeys, about 16%. But in our closer relatives, the catarrhines, so this is a 30-million-year-old group that includes Old World monkeys and apes, same-sex mounting is common, occurring in over 70% of genera and in most or all species of some genera. And many species also engage in other types of non-reproductive sexual behavior with both male and female partners. Like us, these primates interact sexually both for pleasure and for social ends. And their sexual behavior is influenced by complex social and environmental factors. Our evolutionary heritage thus includes a long primate history of multifunctional, multifaceted sexuality with cross-sex and same-sex sexual behavior expressed in a context of underlying bisexual potential. From the diversity of sexual expression across human cultures and history, as well as from recent research focused on sexual behavior rather than identities, it's clear that humans have inherited this underlying catarrhine bisexual potential. Thus, the capacity for same-sex sexual behavior is ancestral. It doesn't need a human-specific explanation. However, we clearly express this potential in different ways than do the catarrhine relatives. One prominent change since our lineage diverged from bonobos and chimpanzees is the emergence of exclusive sexual orientations. This human variation needs a specifically human explanation. So the considerable effort that has been expended over many years seeking biological causes of homosexuality has made clear that, as with most traits, both genetic and prenatal factors have some influence. But these studies don't explain all of the variation. And they've also left the origin of exclusive heterosexuality largely unexamined, often treating it as a presumed ancestral state that doesn't need to be explained, despite the contrary evidence from other primates. So part of the challenge and also the richness of trying to understand human diversity is that as biological potential is expressed or not expressed in different environments, cultural identity categories lump together individuals that differ biologically, and they also split apart others that share fundamental biological similarities. Not everyone who shares an identity, or even a better defined set of behavior patterns or sexual and emotional responses, develop those traits by the same pathway. Even the development of sexual anatomy, which we might think is more simple, it's complex. There are many interacting factors, so that even with well-established statistical patterns, with genetic variants of clear functional relevance, we can't necessarily accurately predict individual phenotypes. Sexual behavior and gender identity are certainly not any simpler. So, what else can we draw on to more fully understand human sexual diversity? What other perspectives can we gain? I think that we can make better use of the comparative context and the growing body of field-based research on animals that naturally express diverse sexual behavior. Research on other primates will help elucidate our ancestral potential, and research on other long-lived species with complex social behavior and relatively slow reproductive rates is also likely to be informative, given that we share some important traits. We can also ask, what else has changed in the last 6 million years or so since our lineage diverged from chimpanzees and bonobos? Of course, there have been a lot of changes, but since the evolutionary challenge in explaining exclusive homosexuality has to do with reproduction, let's look at that. So human reproduction is profoundly different from that of our closest relatives. Mother apes are not out of physical contact with their babies for 6 months or more. They do all of the carrying, all of the feeding themselves. They don't give birth again for 6 or 8 years, not until after their first youngster is nutritionally independent. Human babies are the neediest, most expensive creatures on the planet, and they take the longest to raise to independence. Also, mothers often have multiple dependent young at the same time. How is this even possible? We used to think the solution was the evolution of biparental care. But the highly variable nature of men's commitment to their children is inconsistent with this hypothesis. It's completely different than the extreme paternal reliability that we see in monogamous biparental monkeys. It's now clear that we evolved as what biologists call cooperative breeders, which means mothers and children need and get substantial help from a whole range of others, including relatives, community members, sometimes fathers, but not only fathers and not always fathers. So this kind of help is important in modern industrialized societies, but in our evolutionary past, help would have been essential for keeping children alive. So unlike my frogs, human reproduction and evolution, it's about far, far more than just conception or getting the kids hatched. It's about raising them and making sure at least some of them survive long enough to provide grandchildren. The neediness of young in cooperative breeders makes successful reproduction highly contingent on mothers having adequate social support. And natural selection acts against even trying to reproduce if that support is lacking. The potential evolutionary cost of some family members not reproducing should be considered in this context, because individual reproduction may not always be the best strategy. We see this both in humans and in other species that have independently evolved a cooperative breeding system, dependent on helpers. Their interest in reproduction and even maternal commitment to newborn young, it's context dependent. This uncertainty of reproductive success, the fact that mothers can't raise kids successfully without sufficient help, it substantially increases the evolutionary benefits of helping your relatives, and also, frankly, of exploiting the child-rearing labor of others. It expands the universe of possible family structures, and it generates a lot of creative flexibility and diversity in childcare arrangements. So cooperative breeding created a radically new social environment, inducing and selecting for empathy, and improving our mutual understanding. It shaped the evolution of many human traits that differ from other apes, including our prosociality, our communication skills, and our ability to cooperate in realms way beyond childcare. Arguably, cooperative breeding made us human. Many facets of human variation reflect this influence. Indeed, cooperation can enable specialization and diversification of skills. Did cooperative breeding also shape our sexual diversity? Well, so the US and Europe are probably not the best places to study this, in part because of how Western industrialized societies have changed the structures of family life and of community life. And in part because discrimination against sexual minorities changes the contributions that people can make to society and to families. We may learn more from cross-cultural comparisons, since the way that sexual minorities are integrated into family life varies, and some cultures are, and have historically been, accepting of a greater variety of genders and sexualities. For instance, in striking contrast to results from the US and Europe and Japan, in Samoa, Paul Vassie has repeatedly found, that non-reproductive feminine male androphiles, called fa'afafine and socially recognized as a third gender, are unusually helpful to their families compared with multiple different control groups, and they display avuncular cognitive traits that are consistent with the evolutionary hypothesis of kin selection. He's now doing parallel work with the Zapotec Muxes, a similar third gender group in Mexico. So we still have a lot to learn, both about the evolution and development of sexual diversity, but recognizing the repeated evolution and diverse expression of same-sex sexual behavior in animals, our long primate history of multifaceted, multifunctional bisexuality, and our more recent evolutionary history as cooperative breeding hominins seems important in defining what questions to ask. Ecological developmental biology is giving us new ways of understanding variation by focusing on plasticity and how different subsets of potential are expressed in different contexts. But we cannot understand variation, animal or human, individually expressed or historically evolved, unless we notice it, take our observations seriously, and do the work needed to test hypotheses that emerge from those observations. Some explanations based on familiar subsets of natural variation fail to account for or can even block us from seeing other ranges of variation. When we notice animals or people doing things outside our expectations, it creates new opportunities to improve our understanding. Ultimately, the best answers will work across all of the variation present in nature. So I want first to thank the frogs for just doing what they do, regardless of what anyone thinks they should be doing, and for all of the years that I've spent watching them. I thank my biological family for raising me in Kenya and Canada, so as long as I can remember, I've known that environmental variation matters, and the best thing to do depends on context. I thank my chosen family, especially my partner, Ondine LeBlanc, for always questioning my assumptions and helping me to build a more nuanced understanding of diversity. And I thank all of my students, past and present, and my collaborators for doing science with me, my early mentors for encouraging me to pursue my own ideas, my colleagues in biology and in gender and sexuality studies for so many interesting conversations, my institutions, Boston University and the Smithsonian Tropical Research Institute, and the National Science Foundation for supporting our work. And thank you for coming tonight. would be happy to take questions. I would be delighted to take questions and actually have a couple of more video clips that I'm just going to let play in the background while we do that, because they're too long to put in the middle. But this is another snake video that we just recorded this summer. And I'll just let that play, and I'm happy to take questions.
If you have a question, raise your hand. There are microphones that will come.
Hi, Karen. I was going to ask, how do the chimps, or I forget which species they were, how do they regulate not having kids for 7 to 8 years after they give birth?
Well, so they have lactational amenorrhea first, right? So nursing mothers don't ovulate, and so basically they don't resume menstrual cycling for quite a long time, and they nurse for a pretty long time. Yeah. So even though they're like having a lot of sex, right, and they have these crazy sexual swellings, so it looks like they're, going into fertile cycles, and especially bonobos spend a lot of time in that, kind of hypersexual state. But they do it when they're pregnant. They do it when they're, when they're have babies and they're not actually cycling. They do it essentially when they're teenagers and they're not able to conceive yet. So there's a lot of kind of false advertising of fertility, you could say.
That was great, Karen. So you alluded to, I think, with the drying and things. So I'm wondering, over the years, you and your students studying, many, many years, I guess over 20 years of watching the frogs in the clutches, have you seen any effects of climate change specifically? I'm just curious. But yeah. Yeah,
that's a really good question. So a few years ago, we had an El Nino in the world, but also in Panama. Many of my students were there then. And for the red-eyed tree frogs, basically almost none of the eggs that were laid at the pond that we left at the pond survived. They were just all drying out. So we started even collecting eggs that we weren't going to use just because we had them on our permit and we could keep them alive in the lab by spraying them. So red-eyed tree frogs, the mother puts a lot of water into the clutch initially, and they're not as dependent on rain. The hourglass tree frogs are super dependent on rain. So, they're not very good at being terrestrial eggs yet. And so Justin also looked at weather patterns. So one thing about working in Gamboa is there's a very long because of the Panama Canal, a long record of detailed weather data. And Justin identified the kinds of conditions under which eggs would be laid and then die. And those conditions are clearly increasing, have increased over the last 30 years. So yeah, that's a thing. And they can hatch early to escape from drying, both red-eyed tree frogs, and glass frogs and hourglass tree frogs, but only to a certain extent, right? So they also can, if they dry out too fast, too early, they'll just die. Yeah.
So I just have a question. You mentioned often about the importance of diversity in science. Mm-hmm. I'm wondering if you think our university is doing enough to encourage diversity here, or in the different types of diversity, that if there's enough of an effort here to increase diversity here.
So that's kind of a hard question, because really what is enough? So we definitely are not where we would like to be. We're trying to move in a good direction, and there are multiple efforts underway that are working on that. We all would like, it to be moving faster. Yeah, and it's we recognize that diversity matters, and that different people do ask different questions. And this is I see that in my own lab, right? That's important. But yeah, I think on an institutional level, I mean, maybe that's a question for for President Brown as much as for me. We have many members here of the LGBTQIA task force for faculty and staff to try to improve the working environment here. There are there's a group from the university committee that's working on student issues. And where's Joyce? We're also a pilot institution for a new program from AAAS that is, it's like the Leeds Building Certification, but for STEM diversity in universities. So we are entering into that process, and part of that is a commitment to improve.
Thank you. So, first of all, thank you so much for taking the time to give this talk. It was amazing.
Thank you.
Yeah. As a neuroscientist, I kind of noticed, like, at many points in this lecture, my breathing kind of quickening and, like, my heart rate speeding up, because this is just, like, oh, so cool. But I was wondering if you know of any studies that have been done looking at maybe activity in these embryos, or even in like frog parents when they're deciding whether or not to lay their eggs on land or in the water, that are electrode-based studies or like activity-based studies in the brain. Because I would be really curious about that.
Wouldn't that be so cool? Okay, so my newest PhD student is really interested in what's going on in embryos' brains. And we, I've been working on these frogs since 1991. I've been working on them here since 2001. And there's gradually building up a bigger and bigger picture of them. So my student Julie Jung saw one of her pictures in there of sensory surface in the inner ear. So we now have pretty good evidence that the snake sensor is the vestibular system of the inner ear. We tried to do electrophysiological recordings, auditory brainstem recordings, to see what level of vibration they could sense and stuff, but the embryos are so small and the electrical currents are so weak, we couldn't do it with the field portable EFIS rig that we took to Panama. So it was worth a try, but we're not there yet. But we have in a sense the input from the vestibular system, we have the output of the hatching glands, and now those have to be connected through the brain. So in a few years we may know more about that. The first question, which we might know even in weeks, is, are the hatching gland cells innervated, right? What's the mechanism by which they just dump out all that enzyme so quickly? Maria Jose is working on it. You could talk to her.
Congratulations, Karen, on a wonderful talk. As a neuroscientist again, and we've had this conversation in various contexts, but I thought I'd bring it up here just for fun. The inspiration of your talk is to look at, and in this case, your animals are a non-mammalian system that's given you a lot of inspiration for looking at diversity in your work on human sexuality. But couldn't you also take that same kind of inspiration to acknowledge that in a variety of non-primate mammals, there are really very, very dramatic, well-established dimorphisms, not with basically no overlap in the peptidergic neuron systems or in even some other features of neural sex differences that if we looked more thoroughly and longer in humans might also be found. That we shouldn't just automatically say that because we've done some fMRI studies or a couple of classic neuroanatomical studies using old-fashioned methods, that basically there are no sex differences of any note in the human nervous system.
Well, I would never say that there are no sex differences in human brains, but I think that there's a big difference between something that is a broadly overlapping distribution and a statistical pattern, and something that is dimorphic, so that we have, one set of individuals over here and another set of individuals over here, and no overlap. And we see a lot of variety, right? And I think that it's the other factor, of course, to take into account is neuroplasticity, because how we use our brains, it's very clear, changes their structure. And this has been documented not in a sex or gender context so much, but in occupational context. So like musical training, learning to be a cab driver in London. These things which, you could argue are not as profound lifestyle differences as being raised a girl or being raised a boy in our highly gendered society. Those things affect people's brains. So, we can't do the experiments that you could do on a mouse or a ferret, or I could do on a frog. But I think that the idea that our brains are as dimorphic as our genitals, which even genitals are not fully dimorphic and not fully consistent, I just don't buy that level of differentiation, which doesn't mean that there's no differences, right? Those are compatible ideas.
Hi. I was wondering if in any of your studies of the frogs that had care of the egg clutches, if you then saw in future generations any impact on whether or not those frogs would care for their egg clutches if they hatched early.
So we have not done any kind of cross-generational studies of this. That would be that's not the low-hanging fruit in terms of the amount of effort invested and the kind of possible outcomes of it. In all of these species that we're working with, there's this large amount of plasticity in hatching, and it doesn't it doesn't necessarily mean if a frog hatched early because its father left, that frog should not look after its young, or vice versa, if it had a good dad, that it should therefore be a better dad itself. Because the circumstances, of the next generation's life may be different. And I think what is carried forward is the capacity to respond in a range of ways to these different environments. It was fascinating to me that Jesse found this evolutionary correlation between the amount of plasticity that the embryos show and the amount of care that the good fathers in that species show. And it's not, so we used to think, well, maybe if we have longer parental care, we'll have evolution of the young to spend longer in that cared for environment. That's not what happens. They evolve the ability to potentially stay, but they never give up the ability to leave if they need to, because life is just not that predictable. Bad things can happen to parents, even in the best of intentions.
One more question.
Hi.
Hi.
So I'm curious to know if you've had the chance to look at these behavior patterns over a larger scale, like differences depending on ecosystems or temperature conditions, or looking at mapping incidents of early hatching over a larger landscape.
Well, so we do know that across species of frogs, like broadly across the frog phylogeny, there's 15 different families where it's been documented, but it's not like the same in all of those. Some of those are aquatic eggs, and they're using chemical cues to respond. Typically, the aquatic eggs, the magnitude of a shift in hatching timing is smaller than what we see with the terrestrial eggs, where we have this very clean separation between what's going on in the terrestrial egg environment, what's going on in the aquatic larval environment. And I think, I don't know if we have enough, well, we certainly don't have enough to do a robust phylogenetic test of this yet, but from the general pattern, it looks like when you have this cleaner separation of environmental factors that are selecting on the egg stage and the larval stage, that gives you potentially more plasticity. But that's a good question, and will be interesting to say. That you have to accumulate a lot of data, and each of those data points takes maybe years to get.
I think all of you will agree with me that this was a celebration of Karen's science and her discoveries and of our community. So there are two things I'd like to give her to remember the day by: the poster and an envelope.
Thank you.
Thank you.